The overall goal of this procedure is to prepare AD model mice brains for detection of neuritic plaques using 4G8 immunostaining and thioflavin S staining. This is accomplished by first extracting the mouse brain from the skull and passively perfusing in 4% PFA, followed by submerging in 30% sucrose solution. Cryosection the brains longitudinally to a 30 micron thickness. Next, stain neuritic plaques using the 4G8 antibody and visualize by the DAB method. In addition, stain neuritic plaques by thioflavin S staining and visualize using fluorescent microscopy. Ultimately, results obtained can show neuritic plaque deposition in brain tissues through an antibody-based detection and staining with the thioflavin S dye.
The main advantage of these techniques over existing methods is that 4G8 staining and thioflavin S are highly selective for neuritic plaques containing A beta proteins.
Although this method we shown here is to detect a beta plaque in Alzheimer's disease transgenic mice, this method can also be adapted to detect a beta plaque in Alzheimer's disease human brain tissues.
Sacrifice the transgenic mice using carbon dioxide gas, followed by decapitation. Extract the brain and dissect it longitudinally at the center. Flash freeze half of the brain on dry ice for biochemical analysis. Submerge the other half in 4% paraformaldehyde for at least 48 hours at 4 degrees Celsius. Transfer the hemibrain to 30% sucrose solution at 4 degrees Celsius. Initially, the hemibrain will float in the sucrose solution, but after 24 to 48 hours it sinks to the bottom. Before cryosectioning, mount a thin layer of OCT on the knob and allow it to freeze at -20 degrees Celsius. Remove the cerebellum from the rest of the hemibrain. Place the remainder onto the layer of frozen OCT. Immediately cover it with OCT and allow it to slowly freeze at -20 degrees Celsius. Set the cryostat to section thicknesses of 30 microns and collect each slice into a container with Dolomos solution. Treat each free-floating section with 88% formic acid for 15 minutes. The sections will appear to shrivel. Then transfer each section to a plate and wash 3 times with Triton X PBS for 5 minutes with gentle shaking. Attenuate the endogenous activity with 0.5% hydrogen peroxide in Triton X PBS for 30 minutes at room temperature with gentle shaking. Then wash the sections 3 times with Triton X PBS for 10 minutes under gentle shaking. Block sections with 5% non-fat skim milk dissolved in Triton X PBS at room temperature for 1 hour with gentle shaking. Continue to incubate sections in primary antibody diluted in Triton X PBS containing 5% milk at 4 degrees Celsius overnight with gentle shaking. Wash the samples 3 times with Triton X PBS for 10 minutes. Now prepare ABC solution according to the manufacturer's protocol and incubate sections in the ABC mixture for 30 minutes with gentle shaking, followed by 3 washes with Triton X PBS for 10 minutes. Incubate sections in freshly prepared DAB mixture for 5 to 10 minutes. When a brownish color develops on each section, wash 3 times with Triton X PBS for 10 minutes with gentle shaking. Mount the free-floating brain sections on gelatin-coated slides and air dry. Clear with a series of xylene treatments and mount in Entolin. Visualize the plaques under light microscopy at 40x magnification and quantify mean plaque count per slice for each mouse. Extract brain tissue from mice as shown earlier. Mount 4 to 5 hemibrain sections onto a glass slide and air dry completely. Dehydrate samples in 70% ethanol for 1 minute, followed by 80% ethanol for 1 minute. Next, incubate slides in 0.1% filtered thioflavin S solution for 15 minutes, taking care to protect the thioflavin S solution and stain slides from light. Wash slides with 80% ethanol for 1 minute, 70% ethanol for 1 minute, and distilled water twice. Now mount the slides in aqueous mounting media and air dry in the dark for at least 2 hours, followed by sealing the cover slip with clear nail polish. Store the stained sections in the dark at 4 degrees Celsius. Finally, visualize green fluorescent stained plaques with fluorescent microscopy within 1 week. In our recent study on the efficacy of the anti-epileptic drug and mood stabilizer valproic acid to inhibit neuritic plaque formation, we used the above-described immunostaining and thioflavin S staining procedures to identify neuritic plaques in AD model mice. A typical 9-month-old APP23 transgenic mouse is stained with anti-A beta using biotinylated 4G8 and detected via the ABC method. Neuritic plaques are clearly labeled with the antibody and are indicated by the white arrows. Here, thioflavin S staining detects neuritic plaque deposition in 2-month-old APP23x PS transgenic mice.
Once mastered, this technique can be completed in 3 days if performed properly. While attempting this procedure, it is important to completely fix the brain in 4% PFA and ensure that the brain completely sinks to the bottom of the 30% sucrose solution.
Following this procedure, other methods like immunostaining for tau can be performed in order to answer additional questions like tau hyperphosphorylation and neuronal fiber tangle.